So we're going to stay with the theme of autophagy, and the next speaker is Avi Patel from the Brigham in Boston, and the talk is entitled Regulation of Autophagy in IPF.
Okay. Well, thank you to the organizers and the scientific committee for the opportunity to present today. I'm going to be talking about autophagy and pulmonary fibrosis, so a common theme. And as you've already heard and probably already know, autophagy is a pretty basic fundamental cellular process, so suffice it to say that it's very cell and environment dependent. What I'll be talking about and the work we've been doing is really mostly in fibroblasts. So this is a basic schema of autophagy, which you've just seen as well. It starts with formation of this double membrane structure, which then elongates and then develops the cargo to be degraded. There's fusion with a lysosome, and then the inner membrane dissolutes and the cargo is degraded and then recycled back into the cytosol. So when we began to think about autophagy in IPF, looking through the literature, there are several pathways or processes in IPF that are upregulated or activated that also play a role in autophagy. And specifically, ER stress, oxidative stress, and HIF-1 alpha have all been in the literature shown to be activated or upregulated, and they're also known to induce autophagy. So we hypothesized actually that autophagy would be induced in IPF. And to look at this initially in human lungs, we looked at several autophagy markers, which you've just heard about, specifically P62 and LC3. P62, as you've heard, is a chaperone molecule which attaches to cargo and transports the cargo into the autophagosome. And it serves as a useful marker because P62 itself is degraded in the final step, so that potentially an increase in P62 suggests some sort of block in autophagy. LC3 is a cytosolic protein that upon lipidation gets incorporated into the autophagosome, where it is called LC32, and as you saw, is differentially migrates on Western blotting, so we can tell the activated form. So we began to look at these markers in IPF lungs. So we have here a Western blot showing whole lung homogeneous protein from IPF lung compared to controls, looking again at LC3 levels. LC3-1 is the upper band, LC3-2 is the lower band. And you can see here that there is a slight decrease in LC3-2 in the IPF lung tissue compared to the controls, quantified here by densitometry. When we looked at autophagosomes by electron microscopy, we also saw a decreased number of autophagosomes. So the top two panels, A and B here, are from IPF lung. Panel C is a control lung, and panel D actually is a COPD lung. COPD, a disease in which autophagy has been shown to be increased, at least in epithelial cells. And quantified here, the number of autophagosomes, you can see that in COPD it's significantly high. IPF is much more similar to the control lung. There's very few autophagosomes by EM. We also looked at P62 protein levels, and here you can see that again in IPF there's significantly more P62 in whole lung homogeneous compared to controls. So putting up this all together, it suggests a picture of where actually autophagy is either not induced or inhibited in some way in IPF lung tissue. So we turn to looking at an in vitro model looking at the effects of Tgf-Beta, and these are in lung fibroblasts, and we again looked at protein levels of LC3 and P62. So initially with LC3-2, the lower band here, you can see that there is a slight induction with starvation without Tgf-Beta, since autophagy is induced by starvation. However, there is a diminishment of LC3-2 in a dose-responsive manner with Tgf-Beta. And then P62, we actually found that, again here with starvation, there's a drop in P62 protein levels as autophagy is induced, but at least at the highest dose of Tgf-Beta, there is P62 goes up. So this, it's consistent with a picture of Tgf-Beta inhibiting autophagy in fibroblasts. Looking at this in a different way, and I don't know if this projects well, but we transfected fibroblasts with a GFP LC3 construct that is constitutively expressed. And in the control situation here, you can see a more diffuse expression of GFP. When you starve the cells and induce autophagy, you have formation of these larger puncta, which represent autophagosomes. However, in the presence of Tgf-beta, you have much fewer puncta, consistent with decreased autophagosome formation. Unfortunately, this is cut off, but to look at this in a different way, we examined autophagic flux, which is something Dr. Sanchez just mentioned, because looking at just static LC3 or even P62 levels may not tell you the whole story. For example, if you have a block in autophagy before the final step here, you would have buildup of autophagosomes, and you would be able to measure higher LC3-2 levels, but that does not mean that autophagy is increased because it may not be going to completion. So to look at that, we use Colloquium, which blocks one of the later steps of autophagy, and did a similar experiment in fibroblasts with and without Tgf-beta and with and without Colloquium. And what we're looking at here is the difference in LC3-2 levels with Colloquium and without. And this is quantified here below in the densitometry. So in the absence of Tgf-beta, you have a higher level of LC3 turnover or autophagic flux. But with Tgf-beta, this drops off. So again, another piece of evidence that Tgf-beta is not just inhibiting these markers of autophagy, but actually decreasing autophagic activity. So I think several lines of evidence that Tgf-beta is inhibiting autophagy. So how is it modulating autophagy? Well, as we've heard and as is well described in the literature, mTOR is a major regulator of autophagy, and it has been shown to be induced by Tgf-beta. And this is just a Western blot demonstrating that. You can see here, it's a Western blot for phosphorus-6, which is a readout of mTOR-C1 activity. And in the presence of rapamycin, an mTOR inhibitor, you have a complete decrease in phosphorus-6. With Tgf-beta only at 24 and 48 hours, you have an induction of phosphorus-6, suggesting Tgf-beta activation of mTOR-C1. But this effect is abrogated in the presence of rapamycin. Another potential pathway by which Tgf-beta may be modulating autophagy is this protein called Tigar, which is more recently described in the literature. And it acts through modulating reactive oxygen species and it is able to inhibit autophagy through that pathway. And looking at Tigar in fibroblasts treated with Tgf-beta, we can see that Tigar is also induced by Tgf-beta. And we also found that protein levels in whole lung homogenin from Ipf Lungs of Tigar are also increased. So two potential pathways, at least, by which Tgf-beta may be inhibiting autophagy, mTOR-C1 and Tigar. So to look at an in vivo model, we used the murine bleomycin model, and we treated these mice with rapamycin to induce autophagy. So mice were treated in 4 groups to control a rapamycin-only group, a bleomycin group, and a combination group. And this was a 21-day experiment. And looking kind of at the outcome of fibrosis, we measured hydroxyproline content, and we can see that the combination group, the last bar here, there is a modest decrease in hydroxyproline consistent with a protective effect of rapamycin. And this has been shown previously in the literature in various different models, including the rat bleomycin model, a systemic sclerosis model. So we also then looked back at the fibroblasts to see how modulating autophagy may be affecting fibronectin and alpha-smooth muscle actin as a marker of myofibroblast transformation. And we use rapamycin here to induce autophagy, and you can see here that you have the expected increase in fibronectin and alpha-smooth muscle actin with Tgf-beta under control conditions. However, in the presence of rapamycin, both fibronectin and alpha-smooth muscle actin are slightly decreased. We did look at collagen levels, and we actually didn't find a significant change in the presence of rapamycin. And then this is a Converse experiment. In this experiment, we inhibited autophagy by transfecting fibroblasts with siRNA targeted against 2 autophagy proteins, either Beclin-1 or LC3. And we can see here in a similar experiment, when you treat with Tgf-beta, in the presence of silencing those autophagy proteins, either Beclin or LC3, there is a potentiation of alpha smooth muscle actin as well as fibronectin expression, suggesting that in the absence of autophagy, cells produce more matrix and are more likely to undergo myofibroblast transformation. So in summary, kind of the model we've been looking at is that treating fibroblasts and potentially in vivo as well with rapamycin, which induces mTOR, inhibits mTOR and induces autophagy, you have a decrease in matrix production and myofibroblast transformation, which eventually may have an anti-fibrotic effect. So in conclusion, we have human IPF lung tissue that demonstrates decreased autophagy markers, a Tgf-Beta 1, which is able to inhibit autophagy in fibroblasts. Rapamycin protects against bleomycin-induced fibrosis, potentially through autophagy induction, and autophagy modulates Tgf-Beta 1 effects on fibroblast activation and matrix production. So I'd just like to thank my mentor, Danielle Morris, Ivan Rosas, along with my colleagues, Dr. Lin, Dr. Guy, and Dr. Haspel, who contributed to this project. Thank you. Thank you. No.
Can you hear me? Okay, well, thank you for that talk. As you know, mTOR has been shown by others to be important in fibrosis, and it has a variety of functions that would be consistent with fibrosis, its proliferative effect amongst them. Have you dissected out how important the autophagy pathway is compared to the many other pathways that might be important in this setting?
Yeah, it's a very good question. That's one of the things we've been struggling with, and we're in the process of doing that. I think there's, rapamycin, of course, by inhibiting mTOR has other effects besides inducing autophagy. So one of the next steps, at least in vivo, is to use another method of inducing autophagy that's mTOR-C independent, as well as looking at autophagy deficient mice or autophagy deficient cells and seeing if
next step. Well, good luck.
Right. Thank you.
Yes. So a flip side of that question, another little cautionary tale perhaps, relates to the concentrations of Tgf-Beta that you need to use in vitro to induce these effects. Because if you block two integrins, you can completely inhibit most of the developmental and many of the pathologic effects of Tgf-Beta. Endogenous activation of Tgf-Beta never generates more than the equivalent of 30 to 100 picograms per mil. So if you, in situations where you need to use more than 5 nanograms per mil of Tgf-Beta, it seems unlikely that those local concentrations would ever be generated in vivo. So I think it'd be really important to show in vivo that manipulating Tgf-Beta signaling has impacts on the reduction in autophagy. Because it's possible that very high non-physiologic concentrations of Tgf-Beta have these effects on fibroblasts, but they might not have much to do with what's actually happening in a whole animal.
Right, no, I agree. I think we did see some effects at somewhat lower doses of Tgf-Beta, 2.5 or 5, but still, super physiologic doses. So I think we do certainly need to look in vivo, and that is one of the things we'd like to look at in vivo effects of Tgf-Beta on autophagy.
Hi, thank you very much for the talk. I thoroughly enjoyed it. Can you hear me? Yeah.
Yes, go ahead.
Your LC3 levels in IPF kind of modestly decreased in the lung tissues, but your electron microscopy do not really correlate to these results. Like, there's not much difference between the IPF and the control subjects with regards to the electron microscopy data. So how would you conclude that autophagy is really decreased in the IPF patients? Or do you mean that there's no change? If there's no change, then how would the TGF-beta pathway influence it anyways?
Yeah, that's a good question. I think it's been challenging to assess autophagy in the human lung tissue. I think all we can say is that it's not induced as we would expect it, based on some of those other pathways like ER stretch, where we typically expect it to be induced. So our hypothesis and our our theory is that Tgf-Beta is perhaps preventing autophagy from being activated, and that if it can be activated, it may be beneficial. I'm sorry, I couldn't hear you. Yeah, we think that if Tgf-Beta, if autophagy can be activated, then it may be beneficial in fibrosis, but perhaps Tgf-Beta is preventing that activation. Yeah.
Andy.
Avi, that's really elegant work. Have you also looked at the effects of Tgf-Beta on autophagy in the epithelial? Because those cells are cells that are, we think, have stress, and that if you could increase their autophagy, maybe that would be protected. Is inhibiting their autophagy, that might be part of why they can't handle their ER stress and predisposed to apoptosis.
Right, yeah, so most of our work has been in fibroblasts. We have done some experiments in epithelial cells. We've used human epithelial cell lines and showed, at least looking at LC3 levels, a similar effect. And we also looked at alveolar type 2 cells from mice, and doing a similar experiment with what we did with the GFP LC3. We also found it looked to be that there was decrease in autophagosome formation with Tgf-Beta, but we haven't carried that out further yet.
Can I ask you a question and draw you out a little bit on the sort of homeostatic role that this would play during Tgf-Beta's effect, say during wound healing, where it's obviously playing a central role? What would be the advantage in your mind for Tgf-Beta to inhibit this process during normal wound healing?
Yeah, during normal wound healing, I think there is possibility, as Dr. Sanchez mentioned, that if inducing autophagy promotes intracellular collagen degradation, that in wound healing, you may obviously would not want that. So I think if suppression of autophagy may decrease matrix production or matrix degradation, that might be beneficial in that situation.
So do you think there might be a temporal kinetic to this, whereby maybe in the early phases Tgf-Beta might drive this effect, but maybe later on, as wound sort of healing progresses, this other effect might come? Or do you think these are all occurring at the same time? Because I can see that, degrading collagen is a good thing during wound healing, but initially you do need that burst of collagen to actually affect wound healing.
Right. Yeah, I think that the time course, the time effects would certainly be important, and I think that's one of the things we'd like to look at least in fibrosis in vivo, is to see, how Tgf-Beta and autophagy are playing. For example, in the bleomycin model, we have not looked earlier to see if there are different changes earlier in autophagy.
Okay,
yeah.
Great. Thank you very much. I think we need to move on. Thank you.